This video is about sections 11.1, 11.2, and 11.3. Chapter 11 overall is about the skeletal muscle tissue, differentiating between skeletal, cardiac, and smooth muscle, and talking about how movement, particularly movement of these muscle types, is a fundamental characteristic of all living things. And we're going to be talking about muscles at the molecular, cellular, and tissue levels. So, here in section 11.1, we'll be talking about the physiological properties that all muscle types, skeletal, cardiac, and smooth, have in common. And we'll also be talking about the elastic functions of the connective tissue that is a component of muscles. So our three muscle types, skeletal, cardiac, and smooth, share some characteristics. First, they are responsive or excitable. They can respond to chemical signals and electrical changes across the plasma membrane. They are conductive. Okay, so when we have electrical excitation happening, that excitation can travel along the muscle fiber. They can contract. They shorten when they're stimulated. We call that a muscle contraction. They are extensible. Okay, so they can be extended or stretched in between those shortening contractions. And they are elastic, meaning they return to their original length after the contraction or extension is over. So we've talked about skeletal muscle quite a bit already. We know it's voluntary, we know it's striated, and we know it's either directly or indirectly attached to bones. Striations, when I say the word striations, what I'm talking about there is alternating light and dark bands that end up looking like stripes when we look under a microscope. So we can see here, it literally looks like stripes. We say voluntary because skeletal muscles are under your conscious control. It's a choice to move your skeletal muscles. And I want you to know that a muscle fiber, or a myofibril, is a muscle cell. They can be long. They can be about 30 centimeters long in some cases. Skeletal muscle is wrapped in 3 layers of connective tissue, and we've talked about this previously. So the endomysium is around the myofibril, the perimysium is around the muscle fascicle, and the epimysium is around the entire muscle. We know that tendons help attach muscles to bones, and that these are made of collagen. And we've talked about collagen in the past. It's a little bit extendable, it's a little bit elastic, it can stretch slightly and then recoils back. It's a very tough connective tissue. All right, moving on to section 11.2. Here we're going to be talking about the structure of muscle fibers at the microscopic level. We'll talk about why we see the striations or the stripes that we do, and we'll talk about the proteins that are very important in muscle fibers. Okay, so this slide is telling us all of the different microscopic anatomy of a muscle fiber. So the first thing we have is the sarcolemma, and the sarcolemma is a specific name for the plasma membrane of a muscle fiber. So we know all cells have a plasma membrane. Muscle fiber plasma membranes have a specific name, the sarcolemma. Okay, we know that all cells have cytoplasm, and when we're talking about the specific cytoplasm of a muscle fiber, it has a special name, the sarcolemma. Okay, so myofibrils take up a lot of the space inside of the sarcolemma and within the sarcoplasm. And these are the long cords of proteins that actually do the contracting. We've talked about glycogen. It's carbohydrate storage in the muscles. It's stored specifically in the sarcolemma, and it provides energy for muscle contraction. And myoglobin. So myoglobin is the red pigment that we see in meat. It's also in your muscles, even though, unless you're a cannibal, you don't eat human muscles. Okay, so the myoglobin is related to hemoglobin, and it also provides oxygen for muscles to use. Muscle, skeletal muscles specifically, have many flat nuclei that tend to be right up against the sarcolemma, against that plasma membrane. And this is a little different than a typical cell. In a typical cell, we like to think of the nucleus as being one, usually, and in the middle of the cell. That's not the case with a muscle fiber. Muscle fibers have many nuclei that are up against the sarcolemma. Mitochondria. Muscle fibers have many mitochondria. Mitochondria are generating ATP, and muscle cells use a lot of ATP. Okay, smooth endoplasmic reticulum, smooth ER. When we're talking about smooth ER inside of a muscle fiber, it has a special name also. We call it the sarcoplasmic reticulum. So smooth ER that we find in a muscle fiber, special name, sarcoplasmic reticulum. And it has a couple things that are characteristic. It has terminal cisternae. These are little sacs at the end of the sarcoplasmic reticulum that cross the muscle fiber, like all the way across from one side to the other. And the purpose of these is to store calcium. And that calcium is released and then reabsorbed to allow a muscle contraction to happen. We also have something called T-tubules. T-tubules are kind of folds in the sarcolemma, that plasma membrane, that kind of give access into the cell and sometimes go all the way through the cell. And when we have one T-tubule associated with two terminal cisternae, what we have there is called a triad. Okay, so now we're getting into the anatomy and physiology of muscle fibers. Okay, so the first component is called the thick filament, and the thick filament is made of myosin. Myosin is the protein that makes up the thick filament. Okay, so it looks like this. It kind of looks like a golf club. It's got a long linear section and then this bulb at one end. We call this the globular head, and it's actually two side by side. And we have two chains twisted around here. And these chains end up arranged in a very orderly way. So these heads are arranged kind of in this spiral shape that you can see. We have a section in the middle with no myosin globular heads. Okay, so our thick filament is shown here in purple. It's made of myosin and it has globular heads that are arranged in a helix. The thin filament is called actin. Okay, it is also two intertwined strands, and actin is actually strings of globular protein. So this is like two strings of pearls that end up wrapped around each other. Supporting these, we have tropomyosin. So tropomyosin typically blocks the active site until the active site needs to become usable. And then we have troponin. Troponin is sitting on top of the tropomyosin, and the troponin is where the calcium binds that allows for the tropomyosin to move, that allows for the actin to interact with the myosin and cause a muscle contraction. Titin is another protein that's important here. It has the property of being very elastic. It can expand and then recoil and maintain its initial length without being distorted. Okay, so it runs through the thin filaments, and it's the anchor for the Z disc and the M line. Okay, so it helps to stabilize the thick filament, the myosin filament, and it also prevents the muscle fiber from being too stretched. Okay, so actin and myosin are the two proteins that interact to do a muscle contraction. Tropomyosin and troponin are the things that regulate muscle contraction. So we have muscle contraction when we want to and don't have muscle contraction when we don't want to. And then we have several other proteins that help out. Okay, one of these proteins that's really important is called dystrophin. Okay, dystrophin, if there's a problem with dystrophin, it can lead to several different clinical problems. Okay, so what dystrophin does is it links the actin to the myosin filaments and then links the myosin filaments to the endomysium. So this is how specifically the muscles connect to the tendons. And if there's a problem with dystrophin, one of the things that we can see is the disease muscular dystrophy. So I told you before, and you've read before and seen before, that skeletal muscles are striated or striped. And this striping comes from the very specific alignment and organization of myosin and actin. And we see this both in skeletal and cardiac muscles. Okay, so we have these very specific light and dark bands. We have a central region in the center called the H band, and then the Z disc is the dark striation within the light band. Okay, so A is the dark band, and I is the light band. So here's your light band, your I band, and your dark band is your A band. Okay, and the Z disc is the anchoring point for the actin thin filament. So a sarcomere is the contraction unit for a muscle fiber. Okay? A sarcomere is defined as the space between two Z discs, and when muscle cells shorten, sarcomeres shorten. So the Z discs get closer together as the thin and thick filaments slide past each other. So the thick and thin filaments stay the same size. They slide past each other and overlap more during a muscle contraction. When the muscle contracts and the Z disc distance shortens, the sarcomere shortens, the dystrophin and the other proteins pull on the extracellular proteins. Okay, so now we're going to talk about the different levels of skeletal muscle. So the first thing, the whole muscle itself. So for instance, this could be your biceps brachii in your upper arm. This is the whole unit. Unit is a whole organ. Organ is made of a couple different types of tissues. So we have bones associated, tendons associated, and then the muscle fibers associated. It has nerves and blood vessels, and the whole thing is surrounded by epimysium. Okay. Then within this muscle, we have smaller bundles called fascicles. Okay, so a fascicle is a bundle of muscle fibers, and several fascicles make up a muscle. And a fascicle also has nerves and blood vessels, and it is surrounded by the perimysium. Okay, if we were to pull out one fiber from the fascicle, this muscle fiber is one single muscle cell. It's long and thin. Sometimes we think of it as being thread-like. We talked about the thing around the outside of it being the sarcolemma, the plasma membrane of a muscle cell. It's called a sarcolemma. And then it has myofibrils on the inside, which actually do the contracting. The sleeve that goes around the sarcolemma is the endomysium. Okay, now we're looking at the structure of a myofibril. And a myofibril is made of even smaller things called the thin and thick filaments. The thin filament being made of actin, the thick filament being made of myosin that we talked about just a couple minutes ago. Then we can zoom in even further and we can look at one section of a myofibril and we can see a sarcomere. And we said a sarcomere is measured from Z disc to Z disc during a muscle contraction. The thick filament and the thin filament slide past each other and these Z discs get closer together. And then when the muscle relaxes, the Z discs get further apart. Then we can zoom in even further, and here we'd be looking right here. and we can see the myofilaments. We can see the anatomy of the myosin and the actin and how they can be associated with each other during a muscle contraction. All right, section 11.3. Here we're going to be talking about how nerves and muscles work together to cause a muscle contraction. We're going to look at what a motor unit is. We're going to talk about the junction between a nerve fiber and a muscle fiber. And we're going to talk about how electrical charges across the sarcolemma cause a muscle contraction. Only when muscle is stimulated by a nerve does the muscle contract. It doesn't happen on its own. If we see cases of paralysis caused by poisoning or a severed nerve, we see associated muscle will be paralyzed and will shrink because the nerve is not causing it to contract any longer. So somatic motor neurons, somatic means body, motor neurons. These are nerves whose cell bodies are in the brainstem and the spinal cord, and they innervate skeletal muscles. Okay, so their axons go to skeletal muscles. When I say motor unit, what I mean is one nerve and all of the muscle fibers that nerve is associated with. So the muscle fibers that make up one motor unit, along with the one nerve fiber, these will be throughout the entire muscle. And because one nerve is working on the whole bundle of muscle fibers, they're all going to contract at the same time. We can't select some muscle fibers but not others when they are all controlled by one nerve. So this produces a weak muscle contraction over a wide area. Generally, a motor unit contains one nerve and about 200 muscle fibers. Okay, so some can be smaller, some can be larger. For instance, in your eye and in your hand, things that need very precise control, we might have fewer than 200 muscle fibers. Things that need less control, such as the muscles in your calf, they might have up to 1,000 muscle fibers associated with one nerve. So now we're going to be talking about the junction between the nerve and the muscle, and we call this the neuromuscular junction. So the synapse is the point where the nerve fiber comes together with the target muscle cell. And NMJ, neuromuscular junction, is the acronym that we'll sometimes see. Okay, so we have this space here, and this is the synaptic cleft, is the space between the axon of the nerve fiber and the acetylcholine receptors of the muscle fiber. Okay, so acetylcholine is a molecule that's really important here. We're going to be moving acetylcholine from the nerve into the muscle through this synaptic cleft. And Schwann cells are things that kind of wrap around this complex as muscle being activated by the nerve. So we know that it's an electric thing that causes the muscle fibers and the neurons to be associated and for the neurons to stimulate the muscle fibers. So when we talk about the study of this electrical activity, we can use the term electrophysiology. And we need to have minimum voltage that will stimulate muscles to act. That resting potential is negative 90 millivolts. Okay, so when we move away from negative 90 millivolts, when that distance becomes greater, then we can have muscle cells being activated. This resting membrane potential, this normal amount of charge between the inside and the outside of the cell, is maintained by something called sodium-potassium pumps. And these are a type of active transport where we're transporting things against the concentration gradient using ATP to maintain the voltage distance of negative 90 millivolts. So in resting muscle cell, we have more anions, the negatively charged particles on the inside, and this makes the negative charge be on the inside as compared to the outside. Then we have positive charge in the extracellular fluid outside of the cells. When the muscle fiber is stimulated, the sodium ion gates open in that sarcolemma, and the sodium ions are allowed to flow. This is with their electrochemical gradient, and this depolarizes the cell. Once the depolarization happens and the nerve triggers the muscles, then the sodium gates close and the potassium, the K plus gates open. And the K plus gates, the potassium ion gates, they're partly repelled because both potassium and sodium are positive anions. And this allows the cell to repolarize and nerve conduction to stop. So this is a very quick thing. We have some toxins that can interfere with the way that the nerves send muscles, or the nerves send signals to the muscles. Okay, so things that inhibit cholinesterase, they bind to acetylcholinesterase. This is an enzyme. Remember, enzymes end with ASE. And this prevents acetylcholase from being degraded. So this is resulting in a type of paralysis. You guys have heard of tetanus. Okay, it's caused by the bacteria Clostridium tetani. And when a person gets tetanus, if they, say, step on a rusty nail and this anaerobic bacteria starts to grow in some sort of closed-off wound because the person hasn't had a tetanus vaccine, what happens then is the toxin that's produced by the Clostridium tetani causes glycine that is normally in the spinal cord, stopping the motor neurons. That glycine is released into the spinal cord, and all of the muscles go into spasms. And we have overstimulation of all the muscles. And we call this lockjaw. It'll literally make it so muscles of your jaw can't unclench. Also, it has a big impact on like your lats and your traps, and it will cause a person to like bend backwards once they're very far along in the being controlled by this toxin. We have something called flaccid paralysis. This is where muscles don't contract and the muscles just stay limp all the time. So if you've ever seen a movie where natives in South America are using poison blowgun darts, that's the kind of neurotoxin that we have here. And finally, this one is my favorite. So one of the most dangerous types of food poisoning is caused by the bacteria Clostridium botulinum. Okay, and this causes the disease botulism. Botulism is a result of a neurotoxin, causes very quick, very severe damage to nerves. What it does is it blocks the release of acetylcholine, and that produces a type of flaccid paralysis like we just talked about. And eventually you stop being able to swallow, and then you stop being able to breathe. Your diaphragm becomes paralyzed. What's interesting is in very small and very localized doses, we can take this neurotoxin and inject it as a cosmetic. We call it Botox. When a person wants to reduce the appearance of wrinkles in their faces, they literally paralyze the muscles of the face with Botox, which comes from this Clostridium botulinum bacteria, and in larger doses would be fatally toxic.